Hello, my dear students. I hope everything is okay. Let's finish this part of lecture 11, the non-aqueous titrations. Non-aqueous titrations are performing acid-base reactions or titrations in non-aqueous media or non-aqueous solvents. The samples in this case are two weakly basic or two weakly acidic compounds that they do not react with corresponding acids or corresponding bases in aqueous solutions. It was found that when they are dissolved in non-aqueous solvents, this increases the strength of the titrant and it increases the strength of the sample and also helps to obtain sharper endpoint. A sharper endpoint means an endpoint that changes rapidly and immediately. The color is not moving gradually, but just sudden change from one color to the other. The solvents used in this type of titrations are classified into four main types. The first type are the protophilic solvents. These are basic solvents. The name protophilic, it means it's proton loving. Proto is proton and philic means love or loving species. So they are proton loving, so they are basic compounds. The examples we are studying are N-butylamine, ethylene diamine, dimethylformamide, and acetone. When these protophilic solvents dissolve the acidic sample, of course they are used for acidic samples, the proton from the sample protonates the solvent, interacts with the solvent and converted into a solvated proton that has a positive charge and resulting with the corresponding conjugate base of the acid. It was found that this solvated proton is the strongest acid that can exist in such medium. It's the strongest acid that can exist in such medium. So the solvated proton is the one that reacts with the titrant. The second type of solvents is the protogenic solvents. The name says they are proton producing or they are source of protons, protogenic. So they are acidic in nature, such as formic acid, sulfuric acid, and hydrochloric acid. And of course, we are talking about very pure solvents. They have zero water content. The third type of solvents is the amphiprotic solvents. They can do both actions. They can possess the both properties. So they have protogenic and protophilic properties, such as acetic acid. Acetic acid can act as an acid and can act as a base. It can act as a protophilic or a protogenic. So when we are having two molecules of acetic acid, one of them acts as protophilic and the other acts as protogenic. The protophilic interacts with the proton from the other molecule and forms the solvated proton. And the other molecule is converted into the base or the conjugate base that has a negative charge. The fourth type of solvents and the last type is the aprotic solvents. They neither produce protons nor interacts with them, like chloroform, benzene. They neither accept nor donate protons and they are inert in action, so they help studying the reactions of acids and bases free from the effect of the solvent. We will take two main examples. That's the titration of weakly basic substances and the titration of weakly acidic substances. The first example will be with weakly basic substances, and the example we are taking would be aniline, and this example applies for basic drugs such as ephedrine, and adrenaline. So the sample is basic in nature. In this case, the titrant will be acidic in nature. That's perchloric acid. We have known the structure of perchloric acid before. Dissolved in glacial acetic acid. Glacial acetic acid means that's pure acetic acid or acetic acid free from water. So perchloric acid in glacial acetic acid is called acetus perchloric acid. Acetus perchloric acid. The solvent used in this case would be glacial acetic acid. It's the solvent for both titrant and a sample. The indicator here is crystal violet. The reaction involves interaction between aniline and acetic acid, glacial acetic acid. The proton from acetic acid moves or interacts with aniline to produce the protonated anilinium. So aniline becomes protonated and acetic acid is released in the form of acetate, that's the conjugate base. So if a weak base such as aniline is dissolved in an excess of acetic acid, it would become protonated. So the protonated sample, the protonated aniline, when titrated with a strong acid like perchloric acid, acetic acid may accept its proton. So when perchloric acid is dissolved in acetic acid in this case acts as a protophilic solvent. It takes the proton and is converted into the solvated proton, and the perchlorate ion is released. So in this reaction, acetic acid acts as both functions. It makes both functions. Here it's in this step it's a protogenic solvent, and here it's a protophilic solvent. So it's like that the final reaction is interaction between the protonated acetate and the protonated acetic acid and the acetate ion to produce the two acetic acid molecules again. So as if the reaction is transfer of protons to the solvent and the interaction between the two solvent molecules, one is protonated and one is an ion or devoid from the proton. A complementary example, an additional example, is the titration of halogen acid salts of bases. The example we have is ephedrine hydrochloride. Ephedrine is the base, the hydrochloride is the hydrogen acid salt. So ephedrine hydrochloride is an example of halogen acid salts of bases. When dissolved, the liberated halide ions, that's the chloride ions, are too weakly basic to react with acetus perchloric acid. So this halogen acid salt releases free chloride ions. These chloride ions are too weakly basic to react with perchloric acid dissolved in acetic acid or the acetus perchloric acid. How to perform the titration in this case? We can do it by using a reagent to enhance the strength of the sample. We add mercuric acetate. Mercuric acetate replaces the halide ions, releasing an amount of acetate ions. Acetate ions are stronger base. These are the basic compound, and the final reaction is not between the ephedrine hydrochloride and the perchloric acid. No, but actually the final reaction is between acetate and perchloric acid. This is the final titration reaction that takes place. And the reagent used here to perform the reaction is mercuric acetate. The final example is the titration of weakly acidic samples. An example of weakly acidic samples are phenols and sulfonamides. Sulfonamides are class of drugs that act as anti-infective or antibacterial agents. The titrant in this case would be standard lithium methoxide. Lithium methoxide. A methoxide is an alcohol devoid from proton. So a lithium methoxide is the titrant used in this case. The solvent we are using are basic solvents like dimethylformamide or N-butylamine, and the indicator used here is called thymol blue. The methoxide titrant interacts with the acidic substance or the acidic sample. It gets protonated to produce methanol, and the conjugate base is released. For a simple compound like benzoic acid, the solvent here is N-butylamine. Benzoic acid interacts with N-butylamine that becomes protonated, and benzoic acid is converted into benzoate. Then the solvated proton, this one, interacts with the titrant, that's the methoxide, to release the free solvent again and methanol. So the overall reaction is like a reaction between benzoic acid and methoxide to release the benzoate and methanol as if there is no solvent.